pasado aquí?
A ver si nos concentramos.
The Copita later, I'm happy to introduce Malin Pinsky, who's from the University of California, Santa Cruz, who is the leader of the Global Change Research Group. And Malin, the stage is yours.
Thank you. Thanks so much for that introduction. I've heard about Doñana for many years and read many of the papers that have come out of the Research Institute, but this is my first time visiting, so I'm really excited to be meeting many of you and getting to meet you in person. And reconnecting with folks I've met elsewhere in Europe as well, so that's been really fun. Thanks as well for maybe slightly delaying your Christmas party for this. I'm excited that that'll be after this, so I'll try to keep it relatively brief. So I lead the Global Change Research Group at the University of California, Santa Cruz, where we're trying to understand how marine species in particular, but more broadly, life on Earth, adapts and what the limits to adaptation are in the context of climate change and variability. And we use a range of tools for doing this. We use mathematical models to help set expectations. We analyze large ecological data sets across time and space to understand population and community scale responses, which is most of what I'll be talking about today. And then I won't talk as much about, but we also use temporal genomics, so genomic time series to understand the role that evolutionary responses can play in the context of rapid environmental change. So as I mentioned, a lot of our research has been on marine ecosystems, but one of the other things that I and others in my group have been really fascinated by is how responses in marine ecosystems compare to responses to climate change and variability in terrestrial and freshwater ecosystems. So that's part of what I wanted to present today, some of our efforts trying to compare between the different ecosystems. So the context for the talk are the large fluctuations in and changes in the environment, the abiotic environment that species on Earth cope with. This is one time series showing fluctuations over the last 750,000 years. Many of us are also interested in the rapid changes that are being experienced now as a result of human activities. But one of the interesting things, I think, is that from the perspective of a plant or a fish or an amphibian, these changes through time, while different maybe in rate or somewhat different in exactly which environmental variables are changing, in many ways are also qualitatively similar. There's a change in their local environment, and how are they going to cope with that change? So we have both a basic science question about how do fluctuations in the environment cascade through biological systems, from genes to individuals to populations and communities, and also what are the important feedbacks between these biological scales? Because biological systems are not responding at just any one of these scales, but then there are important feedbacks that are likely to be governing how the entire system responds. So we've started using the term eco-acclimation as a way to try to capture the range of processes from evolutionary responses through behavior, population dynamics, and community reassembly. There are also, as I mentioned, one of, I think, really interesting opportunities here to compare responses across different realms, because the physical environment is very different in marine, terrestrial, and freshwater ecosystems. A chance to also test how effectively our theory applies across these different realms. So I wanted to start out just by introducing and sort of, or maybe reminding all of us what some of the important differences are. Really basic one, different fluid that life exists in, either air or water. So the specific heat of water is 4 times higher than that of air. The ocean as well is also very big. So even though it has absorbed 90% of the excess heat from anthropogenic climate change, it's actually only risen in temperature at the surface by about 0.9 degrees Celsius so far. So it's actually, the ocean has warmed less than the land surface, which has gone up about 1.6 degrees. So historically, that's led to some to say that impacts of climate change are more dramatic on land than they are in the ocean. On the other hand, a degree Celsius of warming in the ocean is a lot compared to the relatively lower amount of thermal variation through time that ocean ecosystems experience. So this figure here is showing across latitudes the seasonality, so just the standard deviation of monthly temperatures, either on the land surface in green, lots of seasonality at higher latitudes on land, or across latitudes in the ocean, for the ocean surface in blue. Much less thermal variation through time in the ocean, at least at relatively higher frequencies. That's mirrored by relatively less spatial variation in temperatures in the ocean as well. So while it's relatively easy for a lizard or a giraffe to move into the shade or move into the sun on land and change its body temperature, there are fewer opportunities for that in the ocean. There are also interesting differences in the mobility of species and movement constraints on land or in the ocean. Relatively fewer constraints in the ocean than on land. Also really interesting, I think less about freshwater ecosystems, so that some of my colleagues work quite a bit in freshwater ecosystems, and it's interesting to think about how the dendritic structure of freshwater ecosystems constrains the mobility options for species in those realms. When you think about movement, it's not just about constraints and barriers, but also about how much energy it takes to move across space. So I want to pause and ask all of you a question as well. So if you were going to choose to move a kilometer, would you rather swim or would you rather run? So who would rather swim? Some hands are going up. Who would rather run? Yeah, me too. I was on my high school swim team, but I find it much easier to run than to swim. And running is what I continue to do now. On the other hand, the buoyancy from water makes it much easier for species that have evolved in the ocean to move than it does for species on land to move. So it actually takes 8 times more energy to run a kilometer than it does to swim an equivalent kilometer. And the drag from being in the water for species that have evolved in the water is actually less of an issue. There are also important differences when it comes to the movement of propagules. So larval fishes or invertebrates can travel tens to hundreds of kilometers carried by ocean currents, much more than even seeds for many plants. So dispersal even at young ages is also greater in the ocean. One of the other interesting distinctions is just the timeline of human impacts in terrestrial and marine ecosystems. Direct human hunting of animals on land was a major cause of extinction 10,000 years ago or more. But now our primary impact is through transformation of habitat. In the ocean, industrial scale whaling started a couple hundred years ago. Industrial scale fishing is a late 20th century invention, especially after World War II. And habitat transformation still remains quite localized in the ocean rather than as widespread as it is on land. So given these differences, I think it raises some really important and interesting questions. Do responses to climate change and variability play out similarly between marine and terrestrial ecosystems? Are similar processes important? And are similar biological scales of processes involved in responses to climate change across these realms? So with that, I wanted to introduce a little bit of the research that we've been doing, trying to dig into differences in individual scale responses and whether they differ across realms. Then move up in scale to think about population level responses, and finally end thinking a little bit about community scale responses. I'll mention some of the work we're doing on evolutionary responses, but I won't get into it. So at the scale of individual sensitivity to warming, it's useful to think in terms of a thermal performance curve that maps out how fitness differs across temperatures. There's a maximum temperature a species can tolerate and a minimum temperature. The physiological mechanisms that underlie these are, these are especially useful for ectotherms. The exact physiological mechanisms often, or they can differ between species, but often involve metabolic rates relative to either oxygen or food supplies. Cell membrane fluidity can also be important, protein structure as well. And then in terms of the tolerance for future warming, a relatively simple but useful comparison is to think in terms of a thermal safety margin. So we can look at the range of temperatures that a species experiences and that the body temperatures that a species experiences, and compare that to the upper thermal limit. And that difference is called a thermal safety margin. It's useful especially when we're trying to compare across a large number of species. So if we first just think about the upper thermal limit, there have been hundreds of, probably thousands of experiments that have been done measuring upper thermal limits across a wide range of ectotherm taxa, both on land and in the ocean. And a number of colleagues and I a few years ago looked at a large assembly of these experiments that have been pulled together. These are looking at a couple different kinds of experiments. They're critical thermal limits that look at when organisms lose mobility, like insects falling off a wall, for example, or a fish losing mobility. They're also the temperatures at which 50% of an experimental population die, so LT50 measurements as well. To make a heterogeneous data set like this more comparable, we can correct for the temperatures at which species have been acclimated. There's something called an acclimation offset. And we've also looked at questions about how fast the temperatures are increased, or the study duration, which has actually those effects, those experimental design factors have relatively small impact on the measured upper thermal limits. So these provide us a cross-realm indication for upper thermal limits across taxa. And we can plot them, again, across latitudes. And here on the vertical axis, we have temperature for the upper thermal limit. Each of the dots is one of those experimental measurements. And then the line is a general additive mixed effects model that allows us to account not only for the experimental methods, but also for the phylogenetic relatedness among species. You can see on land it's relatively flat across latitudes, with some interesting wiggles I'm happy to talk about later. In the ocean, though, we have a stronger drop in that upper thermal limit towards high latitudes. So this is part of what makes a thermal safety margin. The other part is looking at the body temperatures that species are likely to experience. So a lot of researchers use average temperatures, and yet we know that it's the extreme high temperatures that are most likely to have strong biological impacts. So I'm going to show you our results looking at the extreme high temperatures that species are likely to experience in each location. Thinking about body temperature, though, is a lot more than thinking about air temperature, because the body temperature that an ectotherm experiences depends on its exposure to solar radiation, convection from wind, conduction to the surface that it's standing on, as well as its behavior and where in its local landscape it decides to be. So the approach that we took across such a large number of taxa was to look at microclimate variation in the shade at each of the locations that these organisms had originally been collected for the experiments that we'd collated together. So it's an hourly microclimate model run for 20 years, and we look at the high temperatures that the high hourly temperatures that these species are likely to experience. And that accounts for radiation, convection, conduction, and then behavior by looking at the shady locations. So I'm plotting those calculations in orange. Again, each of the dots are for each of the individual studies, and then the average across latitudes in orange. In the ocean, it's quite a bit easier because convection and conduction are very rapid processes in the ocean. Ectotherm body temperatures equilibrate very quickly with ocean temperatures. So we can use ocean temperatures at depth as an indication of the body temperatures for marine species. What's trickier, though, is actually thinking about behavioral thermoregulation. So the approach there was to look at body size, habitat, and mobility mode for each of these species, and from that calculate how likely they were to be able to access deeper and colder water at each of these locations. So from that, we calculated body temperatures in the ocean, again showing you across latitudes in orange. Quite a bit smoother in the ocean. So from the difference between these two lines for each of these paired dots is what allows us to calculate thermal safety margins. So thermal safety margins are displayed here again across latitudes from the southern hemisphere to the equator to the northern hemisphere. Higher thermal safety margins up here. Each of the dots are one of those species calculations. And then the smooth changes across latitudes are green for land and blue for marine. And there are a couple of interesting things that I take away from this plot. One is that the lowest thermal safety margins are actually not at the equator. So some of you may be familiar with, there's been a lot of discussion about species at the equator being most sensitive to future warming. And yet when we look at these thermal safety margins, it's actually species at slightly higher latitudes. So these are the latitudes where extreme high temperatures are even higher than in the tropics. And that's partly because there's less humidity, but also longer summer days heat up more at these latitudes than they do right at the equator. I think some really interesting questions about what that means for tropical species either moving to higher latitudes or whether these species really are most sensitive to future warming. The other thing that comes out of this is that across all latitudes we have less thermal safety for marine species, suggesting at least at this level of individuals, it suggests that they're most sensitive to further warming from this physiological perspective. So just to summarize this a little bit, we can think about a whole range of processes by which species cope with climate change and variability across scales of time. Think about that scaling probably in terms of generation time, or space, probably in terms of body length, which scales with mobility of different organisms. And so we're seeing that from the perspective of physiological tolerance and behavioral thermoregulation, those processes are relatively more important on land, so this green circle, versus what we're seeing in the ocean for the blue circle, just as a way to summarize what I've shown you already. Another important process, though, is acclimation. So the ability for individuals to change their upper thermal limits when they're exposed to high temperatures. So if species can acclimate perfectly, even as body temperature goes up, their thermal safety margin will stay the same. That would be perfect compensation. If they've got 0, then it'll decline 1-to-1 with every degree increase in body temperature. Based on some work that Gunderson and Stillman have done looking across acclimation experiments in the lab, there's not a whole lot of plasticity on average for terrestrial species. It's not that there's none, but there's just not a whole lot. There actually is a bit more in the ocean. One of the hypotheses is that it has to do with the relatively lower availability of thermal refugia and less spatial variation in temperature in the ocean. So we can add these differences in plasticity, though, into thinking about thermal safety margins as future temperatures continue to rise. I guess I summarize this with sort of more physiological plasticity in the ocean than on land. We can add that into projections of future changes, and yet even with this acclimation, we see a bit less thermal safety in the ocean than on land. So I think the take-home message at the level of the individual, at least from this comparison, is that marine species are more sensitive to warming than terrestrial species. This is a relatively crude metric using thermal safety margins. When looking at individual species or just pairwise comparisons, a more powerful way can be to look at energy budget models, and that would be really interesting as a future way to explore these kinds of questions. But thermal safety margins at least provide us an accessible way for looking at this broadly across many species. So if we want to scale up to thinking about populations, based on what I just showed you, we might hypothesize that marine species are also quite sensitive in certain parts of their populations to future warming. So we can think about a population spread across latitudes. There's a warm range edge at low latitudes, a cold range edge at high latitudes. And the processes I've been showing you so far allow species to persist in place even if temperatures increase. On the other hand, if these kinds of mechanisms are not sufficient, then that low latitude range edge will contract towards higher latitudes. So if marine species are in fact more sensitive to warming, we might expect that range contractions at that warmer edge are also more common. So just to show you an example from the northeast US, we're looking at the part of the northeast US coast from North Carolina up to the Canadian border. This is the continental shelf, and this is from a scientific survey that was conducted in the late 1960s. So areas in this map that are everything from light blue up to red are higher biomass densities of longhorn sculpin, a species that's relatively common on the continental shelf. And back in the 1960s, the southern range edge was right near Washington, D.C. By the 2010s, that southern range edge had contracted about 300 kilometers further north up towards New York City, again from a repeat survey that had been done. So we have many examples like that from marine systems, and many people around the world have surveyed low latitude range edges for a range of different ectotherms. So we pulled together those observations for terrestrial and marine ectotherms, looking in particular at studies that had reported results for more than one species because we were concerned about publication bias. That might be biased towards reporting range edges that have moved. So we included studies that also reported stationary range edges. So we went through about 800 papers for marine species, had data for about 100 species, half of which had already contracted towards higher latitudes. The equivalent for terrestrial ectotherms was about a quarter. So actually seeming that higher sensitivity of marine species at the individual level was being mirrored at this warm range edge for marine species as well. One alternative explanation, though, is that marine species are just harder to detect and they're more mobile. They move around more. So we also trimmed this data set to about 70 of the species that had low mobility. And yet, again, about half of them appear to have contracted towards higher latitudes, suggesting that it's also population extirpation, not just mobility. So this combines with other evidence to suggest that across these range of processes, we're seeing greater trailing edge persistence in terrestrial species and relatively less in marine species. This is only part of the story, though, because when we're thinking about species survival, it's not just whether a species can persist in place, but also whether it can expand into new locations and potentially make up for the loss of habitat at low latitudes. So that brings us to this other set of processes, either moving across and expanding across latitudinal gradients or elevations or depth gradients in the ocean. So answering questions about range, contractions, and expansions relies on having data that are spread across time and across space. And there are many really impressive data sets on land, including from citizen science programs like eBird or iNaturalist that are now starting to record observations at very large spatial scales. In the ocean, though, it's historically been quite a bit more challenging. There haven't been equivalent data sets for understanding range dynamics in the ocean. And that's not actually because no one is out there surveying. It's just that a lot of those surveys have been conducted by governments or affiliated organizations and end up stored away in various computers or files and just haven't been accessible in the same way that many of the terrestrial data sets have been. So one particular kind of survey called a bottom trawl survey has been used on continental shelves across all oceans, on all continents, including the subantarctic, around the world, from the Arctic all the way down to the subantarctic. And these are scientific surveys where boats go out, they drag a net near the bottom, and they count and weigh the species that are caught. Primarily focused on fishes, but many of them catch and record invertebrates as well. They're originally designed to support fisheries management, but they're independent from fisheries observations. They're not biased by where the fish are. They're often stratified random designs. But as I said, these data sets and these surveys have been very scattered and very difficult to compare. So in an effort that has been led in many ways by Aurore Moreau, a researcher based in France now, but was a postdoc for a couple years with my group, Aurore pulled together an international consortium of researchers working on similar data sets in many different countries. And it's a project called Fish Globe. We actually just had a working group meeting in Montpellier, France, to talk about this data set and trying to develop it further. And one of the things we've done is published a integrated data set across many of these surveys. There's a data paper that came out earlier this year, if anyone's interested in looking at large community scale data sets for marine continental shelves. Relies on the collaboration of many scientists all around the world. It includes a couple thousand species, many hundreds of millions of organisms that have been observed starting about 50 years ago. Exact date, start date depends exactly on which survey. Some of these data are also displayed in a couple websites. These are animated maps that we've put together, and now actually the National Marine Fisheries Service in the US maintains this as what they call the distribution and mapping portal. This includes many of the US data sets, and an earlier version we put together includes US and Canadian data sets as well. It's been a powerful way to engage elementary and high school students, as well as journalists and other non-scientists. So one of the things these data sets record, though, are the position of species range edges through time. So some of the initial analysis of range edges was led by Dr. Alexa Fredson. She was a postdoc in my group a few years ago, now faculty at University of California, Santa Cruz in ocean sciences. And she was interested in this question about leading edge expansion and trailing edge persistence and how those rates of change compared. She was looking at particularly at 43 species of bottom-dwelling fishes in the Northeast US. And the Northeast US is a part of the ocean that has experienced rapid warming over the last few decades. And so this is something that's called a Hove-Muller plot. And what you see is latitude on the vertical axis, year on the horizontal axis. Color indicates temperature. It's colder up north, warmer to the south. But the lines are tracing particular temperature, the latitude of particular temperatures through time. You can see, for example, the 18-degree isotherm has shifted north, and the same goes for other isotherms throughout the Northeast US. And then we can compare the rate at which species range edges, either at the leading edge, they've shifted north, not quite as fast as isotherms, but about twice as fast as the trailing edges have contracted. So actually that's interesting. It suggests that for many of these marine species, these changing environments can actually be an ecological opportunity, not just a cost. Because they're actually expanding, in some cases, faster than they're contracting at the other end. And more generally, that these shifts in species distributions can be a very effective way for coping with environmental change, climate change, and variability. Also, the other point I want to make is that there's an enormous amount of variation among species. So we're switching geographies. We've switched up to Alaska in the North Pacific. This particular arrow I'm showing you is for a different species, harlequin rockfish. The center of the base of the arrow is where the center of the distribution was in the 1980s. By 2011, it had shifted over here. Over the same period of time, again, this is from a bottom trawl survey, spiny dogfish barely shifted at all. If we look across the species in this region, it's an incredible variety. We can even zoom out to across North America. I'm now showing you the average across species in a region as these black arrows. There's even more variety. On the other hand, a decent amount of this variation across species and across regions is explained by the spatial mosaic of temperatures and changing temperatures that these species have experienced. I showed you in the Northeast US that isotherms are shifting north. On the west coast of the US, where species over this period were shifting south, actually isotherms were shifting south as well. There's a period where that part of the global ocean was cooling. So that's sort of a set of patterns where we've now moved on quite a bit to thinking much more mechanistically about the dynamics underlying species range shifts. And we're starting to use spatial population dynamic models that are fit against those data sets I showed you, but that can infer the temperature dependence or other environmental dependence, either of reproduction, mortality, or dispersal. And the whole team of PhD students and postdocs who have been developing these methods. Jivanta Bandara in particular is working on this for species in the Northeast US, and he's a PhD student in my group right now. One of our early applications was for Atlantic sea scallop, one of the most valuable fisheries in the US, so it was an attractive case to use. If we use a model that has temperature-dependent reproduction, it actually reproduces the temporal dynamics of this species, where the model is in the solid line and the dotted line are the observations from the bottom trawl surveys. Reproduces not only the temporal dynamics, but also the spatial pattern of declines quite well, with many declines observed in the southern part of the distribution and also reproduced by a model that has temperature-dependent recruitment. So we're now trying to make these kinds of methods much more widely available. I think they'd be useful not just for marine species, but across a wide range, pretty much any species that has spatiotemporal observations over a long enough time period. So I think the take-home message from this part of the talk is that I show that with a large icon for marine and a smaller one for terrestrial there at the leading edge expansion. Okay, so that was individual, population. In the last little bit, I want to scale up to thinking about communities. So one of the striking findings, at least for me over the last decade or so, is that when we think about community composition change, it's usually not a question of loss of species diversity, but of replacement of species within a community. And the rates at which species are being replaced across communities around the world differs dramatically, from relatively static communities over decades to rapid replacement, almost complete turnover over even just a few years. What drives those rates of replacement, though, has been much more of a mystery. So one of the largest data sets of community composition through time is a data set called Biotime. Maria Dornelas and many other colleagues have pulled that data set together. It includes both terrestrial, shown here in green, and marine data sets in blue, and some freshwater in red. About 40,000 time series around the world. It's a wide range of taxa, though a lot of these time series are either for birds on land or for fishes in the ocean. So one of the first places I wanted to start was just thinking about, does the rate of replacement, is that related to the rate at which temperatures are changing in each of these locations around the world? So I'm going to show you a figure here where we've got temperature change changing faster to the right and faster rates of species replacement higher on this figure. So basically, dissimilarity through time is increasing faster higher up on that vertical axis. Happy to talk about more details later if you're curious. And what we see for marine ecosystems, at least, is that we are seeing faster rates of replacement in places that are changing temperature faster. Relatively slow rates, around 1% species replaced per year on average. There's a lot of variation. I'll show you that in a minute. Increasing to closer to 2 or 3% per year at higher rates of temperature change. I did say I'd show you the underlying data. There's an enormous amount of variation. This is an incredibly heterogeneous data set. A lot of that variation is likely from imperfect detection. So because communities are not observed perfectly, some of the dissimilarity and some of the increase that we see is from just species being detected one year and not detected the next year, and then continuing on. We see that actually in negative autocorrelation of residuals in these models. The other thing I want to say is that these data sets have an incredible variation in the variance between different data sets. It violates a lot of the statistical assumptions in most of our statistical models. The analysis I'm showing you here has been many years in the making, and almost all of that has been in methods development and methods testing, trying to figure out which approaches do not produce false positive rates of like 80% or more with some of the initial results, some of the initial approaches that have been used with these data sets. The other thing I wanted to point out is that you'll notice there are two lines here, one for warming and one for cooling. Both are really important, at least suggested from this data set. We don't think as much about cooling. And yet, if you look at the surface of the Earth, in this case I'm showing you the ocean, but the same would apply on land. This is a temperature trend over a 10-year period in the early 2000s. And you can see that much of the ocean surface cooled over that period of time. So from the perspective of an ecological community or a single species, cooling is an important part of what they experience, even over decade-scale trends, even though when you average over all the variability around the world, there's a clear increasing trend over decades. So both cooling and warming are important in terms of what species and ecological communities experience. So I showed you marine results already, but we see very similar responses across terrestrial and freshwater. A lot more uncertainty in the freshwater response. there are fewer data sets as well. One of the other interesting and cool things I think about having such a large data set is we can start to look at interactions. And one of the hypotheses about responses to temperature change is that they should be more pronounced where thermal refugia are not available, and less pronounced in places where thermal refugia are more accessible. So in more thermally heterogeneous landscapes, we expect a weaker response to changes in temperature. So we can look at that as an interaction, and we do actually see that in more thermally homogeneous landscapes, we see a stronger response to changing temperature, and in more heterogeneous landscapes, we see a weaker response, both on land and in the ocean. This is measured as the standard deviation of temperatures within 20 kilometers of each time series. The other interesting interaction that comes out of this is an interaction between human impact from more pristine landscapes to more human-impacted landscapes. This is a metric across pollution, landscape, habitat change, and invasive species exposure. And we see what looks like a synergistic interaction where these more human-dominated landscapes are even more sensitive to changes in temperature as well. The other thing I wanted to talk about briefly as I get near the end is to try to think about not just long-term responses to changing temperatures, but what about responses to short-term events? I mentioned early on that it's often the extreme high temperature events that are most important to individual responses, but that may also play out at the level of communities. And at this point, there's been, the scientific literature, at least on the marine side, is starting to be filled with a really impressive array of dramatic responses to what are called marine heat waves. Similar literature developing on land as well. But where these can be events from days to months, where temperatures are above the long-term averages. And they've been associated with die-offs of kelp, of corals. I'm showing you a picture here for whales entangled on the west coast of the US. Their prey had moved inshore at a time of the year where there are lots of crab fishing that's also happening near the California coast, and a lot of whales got entangled during a marine heat wave. So sometimes very unexpected impacts as well. So we are interested, though, while we have lots of case studies, can we start to generalize across which kinds of marine heat waves have dramatic ecological impacts? So if we go back to the fish globe data set that I showed you earlier, so these are continental shelf ecosystems looking at marine fishes, where we have population abundances and biomasses through time. We can look at whether the biomass either increased or decreased after a marine heat wave. And I'm showing you results across the cumulative intensity of degree days. So how far above the 95th percentile of historical temperatures are the temperatures during a particular heat wave. And we see after a number of marine heat waves, there were declines in community-wide biomass, and that lines up with many of the published papers describing these large heat waves. I'm showing you a very biased subset of the data of the results, though. Okay, showing you the axes. So when we look across all the heat waves that affected these ecosystems in the fish globe data set, though, we see a lot of variation across these ecosystems, including large increases in biomass after some heat waves, such that there's not a common response across different heat wave events. So we're taking a different approach here. Rather than saying there was a heat wave, was there a response in a particular case, and then let's study that particular response. We've instead gone through all the heat waves that were covered by the fish globe data set and asked whether there was a common response across each of those heat wave events. So that's looking at biomass. We've also looked at this, as you can imagine. We thought for sure there would be a common response. We've looked at many different responses. So changes in the relative abundance of warm adapted or cold adapted species, changes in the depth of these species, changes just in the magnitude of change. Was there just a large change in abundance or biomass after a heat wave, whether it was up or down, and no consistent responses so far. So I think a lot of actually interesting questions remaining about what makes some heat waves very impactful and others seemingly not impactful at all. So take-home message from this part is just that in contrast to actually some differences between realms at the individual and population scale, we're actually seeing widespread species replacement across different realms, both marine and terrestrial, in response to changes in temperature. So I guess more broadly, I see this as a few small steps towards trying to integrate our understanding of climate responses across realms and across processes. And I think one of the striking things is that the scales, the biological scales, and at which responses are most important seem to be different across different realms, with these individual scale responses being really important on land. I showed you some results suggesting that from this cross-species synthesis of thermal safety margins, to then these population scale responses being especially important in the ocean, an environment with fewer barriers, greater mobility, but I showed you results there from analysis of large spatiotemporal data sets. And then across all species, all realms I should say, species replacement actually being an important response that's playing out across realms. One of the big unknowns is the role of adaptive evolution. I did mention briefly that my group also looks at genomic time series. I'll just put this here more as, if you want to ask me later about it, I'm happy to talk about it. We're looking at data sets, historical specimens that were collected about 100 years ago from the Philippines, and by a fluke of scientific history were stored effectively in high-proof Rome and have been sitting in a museum in the suburbs of Washington, D.C. for the last 100 years, working with a bunch of amazing Filipino collaborators to redo these collections and understand evolutionary change over the last century in the Philippines. I guess the broader point I want to make is that I think these diverse set of approaches are really important because species and communities are responding to climate change and variability not just at one particular biological scale, but across these range of eco-acclimation processes. So a lot more to be done to understand the feedbacks and the relative importance of these processes across different species. We have different processes likely having different importance across the different realms. And I think if we just focused on one particular biological scale, we'd miss some of those important differences. And then finally, my group does quite a bit of work collaborating with conservation groups and with fisheries management agencies. And I think part of why thinking across these scales is important is that we risk giving the wrong advice if we don't understand how species are coping with climate change, variability, and change. Happy to talk about that part later as well if you're interested. So none of this would be possible without an incredible range of collaborators, the funders that make all this possible, and the many members of the Global Change Research Group over the years who make this so much fun. So thanks for listening.
Thank you, Malin, for a fantastic talk. We have some time for questions.
Thanks a lot. I work actually on human health, and there's also a lot of work on the thermal upper adaptation limit for humans. And one factor, of course, that is important for humans is evapotranspiration, because, of course, we cool through sweating. So I was wondering whether for mammals, this is, I guess, also important, and whether you have taken into account something like relative air humidity in your work. Yeah. That's one question. And my second question is on the results you have on the marine heat waves. I was actually wondering whether you also looked into food web relationships, because I was just wondering whether maybe it's the prey that is affected, and this is why you're seeing like these very heterogeneous results. Yeah. So these are my two questions.
Both are really good questions. So the work I showed you earlier was just for ectotherms, so it avoided endotherms. It would be really interesting to think about doing that kind of comparison with thinking about thermal neutral zones. And We did account a little bit, not for evapotranspiration, but for the fact that amphibians, evaporation is an important part of how they cope with that. So the microclimate models that we ran for amphibians were a little bit different, so that we could include evaporation as an important process. I could go back to the figure, but they were shown in a different symbol, just to point out where we did those calculations differently. I didn't explain that. And your second question was about prey. So that is one of the hypotheses, that it's not just about temperature and high temperature, but about the coincidence of high temperature and either low oxygen or high temperature and low prey availability. Yeah. So we haven't done that analysis yet, but I think it's a really good hypothesis.
Okay. Thank you for your interesting widespread talk. Anyway, yeah. One suggestion question, I don't know, I'm not sure what my comment no? is regarding the distributional shift in fish communities. Probably you have more fresh literature, but I think that fish communities exhibit a contrary Rappaport rule. What I mean is that equatorial communities of fish have a wider distribution, geographical distribution than temperate or high latitude fish. So the question is, in this sense, what's the opinion or your opinion or the prediction we would expect regarding terrestrial, and because terrestrial Rappaport say that higher latitude mammals or any terrestrial organism have larger distributions, and regarding global warming and all the effects and so, what's your idea about this?
Yeah, I mean, marine species distributions are often very widespread. I think that lines up well with relatively fewer barriers to dispersal, relatively larger dispersal abilities for marine species. And so I think that fits in with sort of the broader point I was making about their ability to shift their distributions as well being relatively easier in the ocean than on land.
The temperate or high latitude fish cannot cross the equator in some way because they have not high thermal tolerances than equatorial. This seems a barrier for them. That's one way to explain why high latitude species. But in a context of world warming, what will happen that if equatorial lineage or groups of fishes that have from northern latitude, but originally from tropics, for instance, may have more possibilities to disperse. I don't know.
Yeah.
Is for the possibility of analyze this.
Yeah. We do have, so the data sets I showed you do, in many cases, we have to stitch together multiple surveys to get the full range of one marine species. And it does mean that for many low latitude species in the tropics, they're able to expand towards higher latitudes. Though for species that are already at very high latitudes, they're running out of habitat space further. So it's a question of how fast does that lower latitude, that warmer range edge contract. Yeah.
Okay, thanks.
Yeah.
Thank you very much for your talk. It was very interesting, and maybe I missed the explanation, but you have one of the first figures that you show. It was like related to temperature, latitude, and at some point in the marine ecosystems, you said that it's like a drop in the thermal limits. So
maybe looking at thermal safety margins.
I think it was before that. So you showed the terrestrial, and then you showed the marine. I think it was before that. Was one of the first ones. Yeah, here. So why would you say there is these drops in the thermal limits in marine ecosystems compared to the land ecosystem?
I guess I was just thinking relatively flat across latitudes on land, whereas declining towards higher latitudes more strongly in the ocean. And that's been, I guess that's been described, not just in this particular data set, but from previous researchers as well, that high thermal tolerance limits tend to be fairly flat on land across latitudes. There are some interesting, we do see some evidence of local adaptation, with higher extreme temperatures at 20 to 30 degrees north and south coming out here, and also coming out in the upper thermal limits appearing as well, but they don't fully compensate for each other, which is why we end up with slimmer thermal safety at 20 to 30 degrees north and south on land.
Okay.
If that maybe starts to answer your question.
I don't know.
Okay,
I think I didn't get it very
there
well.
are lots of interesting things to dig into.
I mean, my main question was why the extreme thermal limits in the ocean were very low compared to land. The street, like.
Yeah. Like why? Oh, why?
Why?
I can
Why?
speculate.
I mean, yeah. Why would you say that? Why would you? What is your opinion about that?
Yeah. I mean, to me at least, it implies a couple potentially different things. One, that high temperatures, at least sometimes, are still being experienced on land, and so there's a benefit to maintaining a high upper thermal limit. Or the cost of maintaining a high upper thermal limit is quite high in the ocean, and it's not worth maintaining that.
Okay.
So it would imply a trade-off.
Okay. Thank you very much.
Yeah.
Thanks for the great talk. So when comparing terrestrial and marine ecosystems, it's a mixed bag of species in each bag, right? And totally. So phylogenetically, they're completely, mixed bag. So it always kind of baffles me that there are still, despite that, there's still patterns, and different patterns,
right.
Because such a mixture of phylogenetic lineages that you wouldn't think a priori that there would be a pattern. There's also, if I'm not mistaken, being published that there's different diversification rates for both realms, and it's much lower diversification rate normally in marine species. Yep. So I was wondering, how if you think that that ties up somehow with the population dynamics that you were mentioning. And also in terms of a community turnover, I guess there are also different timescales, but wouldn't you expect then a greater turnover in terrestrial environments? But I guess maybe the timescales are very different. But so is there, do you think, something fundamentally different in marine species that affects not only rate of diversification at an evolutionary level, but also the adaptive or the changes in the distribution patterns compared to the land ones?
Yeah, that's interesting. I do think there are some links there because, one of the explanations that's been put forward for the lower rates of diversification in the ocean is that there's just much more gene flow across space in the ocean. And that greater dispersal ability is also, though, playing out when we look at shifts in species distributions, very different timescales, and also sort of evolutionary process of gene flow versus an ecological process of range shift, but similar underlying cause. We have been collaborating with Dan Roboski and one of his graduate students, trying to link the spatiotemporal dynamics we can detect in these data sets with macroevolutionary diversification rates to see if to test whether the metapopulation patterns are also linked to the long-term diversification rates. We've put together a framework for how to do that, but don't see a strong link.
Do you think, in addition to gene flow, do you think there's any fundamental difference in, for instance, the effective population size in marine versus terrestrial?
Great point. Yes. And actually, sometimes a picture I show is, so the most abundant vertebrate is the domesticated chicken. But in the ocean, the most abundant vertebrate is about, I think it's 100 times larger. Anyway, it's a mesopelagic fish. So effective population sizes tend to be much larger in the ocean. Thousands to tens of thousands are probably even higher. So strength of selection, stronger in the ocean. Strength of drift, much weaker in the ocean. And with less drift, also less chance for vicariance and divergence. Yeah, that's a good point.
So I'm not so familiar with marine system, but I was wondering what about the depth? Does that enter into adaptability shifts and so on?
Yeah, that's a really good question. And we've looked a little bit at depth shifts. I mentioned briefly that latitudinal shifts are partially explained by isotherms moving north or south. The depth shifts are as well, though not as fully. But there are some interesting cases like Gulf of Mexico. Let's see if I can get a good, maybe I can show it here. Anyway, so Gulf of Mexico right here, they can't shift north, but they can shift deeper. And so actually species in the Gulf of Mexico, despite it warming, have been shifting south and moving deeper. I will say the data sets we've been using, these bottom trawl surveys, are for benthic species, so they're on the bottom. We're not able, from these data sets at least, able to look at vertical habitat use. And for some species that probably is important, though less important for the species we've been looking at. Yeah, it's a great question.
Any more questions?
No?
That was very interesting, thank you. Something that really caught my attention was where you showed the many different arrows of the distributions of the fish just south of Alaska shifting in many different directions.
Yes.
And so to the community aspect of that, So I guess that that maybe has some larger impact on how these species are interacting, or I assuming that at least some of these are predators and that there must be some interactions between these species, or how does that have an important impact, or it doesn't matter? Or?
It's a really good question, something I've been really curious about as well. These are largely from a similar trophic level.
Okay.
So, more likely competitive interactions. One of the other interesting things, though, is that these fishes, they all start out small, and then they grow to different maximum sizes. And most of the predators can eat pretty much anything else when it's young. So they do feed on each other as well, despite being from a similar trophic level. Food webs in the ocean are often size structured.
Okay.
They can gate limited is sometimes what we say, but yeah. So really interesting questions about changes in species interactions, and we haven't dug into it yet. I would love to. Yeah. I also don't want to hold people back from the holiday parties. I'm happy to continue.
Are there any more questions? If not, thank you all so much for coming. Thanks a lot for this talk, and see you later at the Copita.
Thank you.